PCR is a series of reactions which make multiple copies of DNA. It is a revolutionary technique used in molecular biology to amplify a single piece of DNA and make thousands and millions of copies of DNA. This technique is invented by Mullis in 1983. Later on, he was awarded Nobel Prize in Chemistry in 1993. This technique makes easy molecular biology. PCR is basically a process which amplify or copy a single piece of DNA again and again to make it absorb visually. This technique is based on vector replication which carries out by DNA polymerase. DNA polymerase is an enzyme which amplify or copies DNA, so DNA polymerase amplify DNA again and again and make multiple copies or produce. This technique is known as DNA polymerase. Three basic things in PCR: PCR design, reaction in PCR, procedure in PCR. If you know these things, you will know all about PCR. PCR design. PCR is a machine which have just a heat block. Thermal cyclers is in a heat block which have ability to change its temperature instantly. Reactions in PCR. There are some components which is required for carrying out a PCR reactions. Template of DNA, deoxynucleotides, phosphates, DNTPs, ACGTs, primers, and tag polymerase. Templates of DNA must be isolated from a cells. DNTPs, you need DNTPs for making a new strand of a DNA. When DNA split, these DNTPs will bind to a single strand and make new copy of DNA. Primer is 15 to 20 nucleotide sequence of DNA which is designed for a specific gene or functional sequence of DNA. Without primer, DNA polymerase cannot bind to DNA and start polymerization. We must have no primer for amplifying sequence of DNA. Tag polymerase. The whole concept of PCR is come from this enzyme. Tag polymerase play a basic role in PCR. This enzyme was isolated from hot spring bacteria which have capability to amplify a piece of DNA. All these things are the basic requirement for amplifying DNA. Now procedure. How they works. PCR amplification cycles consist of three basic steps: denaturation, primer annealing, extensions, or polymerization. Denaturation. First step, the template is heated at 94 degrees C for 1 minute or up to 5 minutes. At a high temperature, the DNA becomes double strands into a single strand of DNA. Primer annealing. In second step, the two primers, forward and backward primer, is binds to the complementary strand of single strand DNA by lowering temperature up to 54 degrees C approximately at required 2 minutes. Extension or polymerization. Finally, step the tag polymerase binds to the primer and synthesizes new DNA. DNTPs binds one by one and take 1 minute at 72 degrees C. These cycles are going again and again and amplifying a new strands of DNA, and these strands of DNA the PCR make it thousands and millions of copies of that strand of DNA.
که تاسو زموږ د فیسبوک او انسټاګرام پیج ته راتلل غواړئ د هغې لینک په ډسکرپشن کې دی. که دا ویډیو ستاسو خوښه شوي نو دا ویډیو ضرور لایک کړئ او کامنټ وکړئ او شیر کړئ دا ویډیو د خپل ملګرو سره او سبسکرایب کړئ زموږ د یوټیوب چینل. د دې سره به تاسو نوي شیزونه وپیژنئ او ورسره بیل آیکن هم وایئ. د دې سره به تاسو ته پته لګي چې موږ نوې ویډیو کله اپلوډ کړله. او په آخر کې د کتل لپاره ډیره مننه.